right, we're live. All right. Welcome to the second installment of the semi-short format Fitomics talk here, where we will focus on a area of research and really dive into one specific study. And in this talk, we will be talking about a paper on neuromuscular adaptations and skeletal muscle hypertrophy. And the title of this paper is Action Potential Amplitude as a Non-Invasive Indicator of Motor Unit Specific Hypertrophy from Pope et al. In 2016. And I am Dr. Daniel Lawson, joined with the co-host and CEO of Fitomics, Dr. Cody T. Hahn. And we are joined with one of my very good friends and colleagues, Dr. Alex Olmos. We did our PhDs together at Oklahoma State University. We were in two different labs, but I was your step lab brother.
Mm-hmm.
Your lab adopted me, and I appreciate that very much. We've published together. We have two papers. One's currently in review, another that you submitted, several more on the way. The three of us are going to be collaborators on a current meta-analysis that we're all working on. So Alex, tell us a little bit about your area of expertise with neuromuscular research, your experience through your PhD, just a little bit of background on you.
Yeah. Well, first and foremost, thanks for having me on here. It's always good to hang out with friends and colleagues. Yeah. So my research specifically looks at how the brain and the muscles communicate, and then how they adapt to various interventions or just life in general. So for instance, during my master's, I looked at the effects of aging on walking parameters and neuromuscular function. Neuromuscular is such a broad term. You know, you go anywhere from looking at electromyography, which is a electrical signal over the muscle that comes from a sensor that's placed over that muscle, that looks at the excitation, right, when we're activating our muscle. I think the general population knows that as a muscle activation, although the term isn't necessarily correct. It's more input excitation or muscle excitation, right? So during my master's, I looked at the effects of aging on walking parameters and neuromuscular function, and we found that as you get older, your muscles are smaller, which makes sense, and they're less powerful, and they require essentially more excitation to complete a relative task compared to a middle age group. So within a 20-year difference in age. So we did that for my thesis. That was a fun, hard project. And during my PhD, we did multiple studies that looked at motor unit properties, and we'll get into that in a little bit. But one of the fun studies that we did was my dissertation, where I examined if you have a group that focuses on getting strong and then another group that focuses on getting strong and bigger, so hypertrophy, does that additional hypertrophy result in greater strength gains? And then do we see divergent motor unit properties because of the specific type of training? So specifically, are there specific training adaptations that influence the way our motor units operate? And so we did a couple of different things. We looked at motor units. We did transcranial magnetic stimulation, where we stimulated the cortex and essentially looked at the cortical excitability. We stimulated the spinal cord, so looked at that excitability, inhibition. So anywhere from the brain down to the peripheral over the muscle. That's my area of expertise, but my area of interest. That in muscles hypertrophy, which is always fun.
Right on. Yeah, and so the transcranial magnetic stimulation, TMS, is interesting. Without getting too far into the weeds on that, it's not an electrical stimulation, but it feels like it. And I was a test dummy for some piloting that we've done, as well as a participant in a study And it's a very unique sensation when you have your head and spine stimulated.
Okay.
And, I like to go into the lab early mornings and crank that thing up and stimulate my head several times until I was cross-eyed to get prepared for the day. No, I didn't do that, but I always wanted to. But yeah, so it's interesting the way that we measure neuromuscular responses and adaptations during exercise, and I guess specifically resistance training, where we would have a participant hooked up to an isokinetic dynamometer, which is like a big leg extension machine, right? And it allows you to control pretty much. It's a commonly used device in a lot of studies. And it allows you to control a lot of different variables, specifically doing like a single leg extension, where you could control the tempo. You can have it dynamic or at a set tempo, and the resistance, right? There's a lot of things that you can set for the protocol. And typically people would get hooked up to sensors. I'm speaking from a less experienced viewpoint than you, Alex, but I do recall that there was a lot of computer screens.
A lot of computers, a lot of sensors, wires.
We had like 5 or 6 screens with different things going on. And you would ask me something about something, and I would look at the screen and I would see a lot of colorful squiggly lines. And anyways, moving on. There's a lot of work that goes into this. I think that's what I want to preface this discussion on this paper in particular, is the amount of work that goes into neuromuscular research is substantial, and it's kind of a black box, I think, no pun intended, because the signals go into a box, and we go through decomp, right? We decomp these signals through an algorithm, and we'll talk more about this in the paper. It's just, I just wanted people to appreciate, as we get into this, the amount of work and some of the unknowns with some of it. So all of that to say, let's talk about this paper, because this is a very interesting paper, I think.
Oh, yeah. It's kind of one of the staples for our lab. So, yeah.
Yeah. And so this is the PI, the corresponding author on this paper was Dr. Jason DeFreitas, who was one of our professors at Oklahoma State University. I believe he has now moved on to Syracuse.
Yep.
He's the department chair there. So he was on your committee, your dissertation committee. And Garrett Hester, was he your mentor during your master's?
Yeah, he was my mentor during my master's. So
yeah. And he's the second author on this paper. So there's a lot of really interesting stuff to talk about with this paper. And I think before we even get into this, I think it's maybe better if we start, Alex, talking about some introductory concepts to neuromuscular physiology.
Yeah.
So if we could just start, what is a motor unit?
Yeah. So is it okay if I share my screen really quick?
Absolutely.
Luckily, I put in a awesome presentation for us, so that when we're ready, I'm going to. Oh yeah. So you got everyone see the screen?
Yes.
Yes, sir.
Cool. So a motor unit is the alpha motor neuron that's located in the spinal cord, and the specific fibers it innervates. Now, the fibers are located in the muscle, and they're kind of spread around, so it's not all clustered together. And so the way we essentially evaluate a motor unit is based on the waveform and the size of the action potential from those motor neurons as they activate the fibers themselves. And so to do that, we place sensors over the muscle. All right. You can do this intramuscularly, where you put a wire, fine wire or needle, and then when you contract the muscle, it starts to produce electrical signals that indicate that the muscle itself is activating. And each motor unit has its own distinct waveform, which is really neat because that's how we distinguish the different motor units. And we see that when we recruit motor units, we recruit them in size, right? This is Henneman's size principle. So typically an early recruited motor unit is typically smaller. The fibers they innervate are smaller. And as we produce more force, we recruit larger and larger motor units. And those motor units are bigger in size because not only is the alpha motor neuron bigger, but the fibers it innervates are also bigger. And so what we do is with that sensor, whether it's in the muscle or over the muscle, we get this electrical signal called electromyography, which is essentially a raw EMG signal. And then we put that signal into an algorithm that you can either decompose yourself, or you can use a third-party software that decomposes it for you. And so what it does, it tries to find essentially repetitive waveforms that are occurring during that contraction, and it identifies spike trains. So it finds the specific waveform and it identifies every time it fires, right? Or each time the motor unit fires. And what's neat is that each motor unit has a very distinct waveform, and then it has its own firing capabilities. And so we see that, let me actually be able to change here. When we start to produce force, we recruit motor units, in a linear fashion, sometimes non-linearly, but an earlier recruited motor unit typically has a higher firing rate. And as we recruit additional motor units that are bigger, but they tend to have a lower firing rate than the motor unit beforehand. So when we get to the highest threshold motor units, which is recruited later on in the force contraction, it typically has a very low firing rate. And so this is actually extracted from one of my papers. And what's neat is we can actually see the relationship between the motor unit size, so the action potential waveform, and its recruitment threshold. And we see that early recruited motor units typically are smaller. And as we recruit, they get bigger. All right. And later recruited motor units, higher threshold motor units are typically the biggest. All right. They should be. We can kind of see that in the top right, where we compare the first motor unit compared to the 20th motor unit recruited. The waveforms are distinctly different. So later recruited are larger, early recruited are smaller. But because of that, we see that early recruited motor units typically have higher firing rates than lower or later recruited. So that's typically how we see how motor units operate. They're recruited in an early or in a orderly fashion, and then they have an inverse relationship between the firing rates and the recruitment thresholds. So it's kind of just the very general understanding of how motor units operate.
Yeah. And Alex, so because this will be, if you actually want to pull that back up real quick, that third slide, because this is pertinent to the methodology of the study we'll be talking about here, is when we're trying to measure these motor units, the protocol is we have MVC, which is maximal voluntary contraction. Yep. So it's the amount of force that the individual's producing. So when we have them in that isokinetic dynamometer, like a biodex, and they're doing a single leg extension, they would do what's called like a tracing of the trapezoid.
Trapezoid tracing, yeah.
And so that would be 50% typically, right? There's different percentages, I guess.
Yeah, it's dependent on what you're trying to target. So, for instance, in this image here, right, their target goal is 50% of the maximal voluntary contraction. And Dan, if I can, Typically, the way we assess motor units is isometrically. Because what happens is when you place a sensor over the muscle and then you move the muscle, the signal starts to move away and closer to the muscle. So it distorts the signal itself. So we can't really truly say this is what's happening. So to kind of avoid that, we do isometric contractions so that the signal is constantly placed underneath that sensor, right? The action potentials or the excitation that's being emitted. But yeah, you can do any percentage of your MVC. We just typically choose 40, 50, 70 because we can really distinguish the different types of fiber types that we're recruiting and so forth.
Right. Yeah. And I think that's a really key point that you mentioned there is the isometric contraction. And so we have multiple sensors that we would put on a participant's leg.
Mm-hmm.
And if we were to do this dynamically, where the leg would move, which there's a lot of studies that report EMG data during dynamic exercise. And it's important that when we're trying to measure and identify specific motor units, that we normalize that to something.
Yeah.
And an issue with making inferences about motor unit recruitment during dynamic exercises is because the sensor doesn't move. It's on the surface level of the skin. It's over the quad. The muscle's lengthening and contracting. It's getting shorter. And that motor unit where it's innervating those muscle fibers, that can shift, right? That's what you're saying. So it can shift and potentially you might see this increase in the motor unit action potential, which the abbreviation on some of these figures is MUAP typically.
Mm-hmm.
And the issue with that, right, if you would agree with this, is we're not confident that we're measuring the same motor unit because it could be shifting underneath the surface.
Sensor,
yeah. So I think that's important.
Yeah. So we see that, like, let's say, for instance, you have a large waveform, right? Large action potential, and that's motor unit 1 or 2, whatever, it doesn't matter. But as the muscle lengthens or contracts, right, the fibers themselves are shifting away from that sensor. And so when that signal, it's still transmitting longitudinally, right? As the action potential travels on both ends from the innervation zone. That signal, it's going to dampen. And so it still has the same waveform, but the amplitude, the MUAP, I like to say, starts to decrease. And so you're not confident that that's the same motor unit from the initial position because of that. So yeah, that's the best way of putting it. Yeah.
Yeah. And there's definitely some implications with that relative to muscle hypertrophy exercises that would be better based on the action potential. We don't have to get into the weeds of that, right? But that's an inherent limitation that we couldn't confidently say that because we changed the adjustment of someone's foot positioning on this exercise during a dynamic movement and we measured a larger MUAP, motor unit action potential, that would translate into it being more effective for skeletal muscle hypertrophy. Yeah. Because on the social medias, sometimes that seems to have been something that some people would use as a proxy. And so to circle back, it's really important to normalize these signals. One is doing it isometrically, and then normalizing it to a 1RM isometrically is important. So just wanted to clarify.
Building off of these points, my friends, Dr. Alex, do you think that, and you differentiated between surface versus indwelling. Do you think the indwelling electrode or sensor placement is appropriate for dynamic contraction and characterization of the motor unit?
Yeah. Yes. The problem is you can only really identify like 2 to 4 motor units, right? And so typically what we do is we have this group, there's this pool of motor units, and we like to regress them to their recruitment threshold, right? That's how we can build that nice, pretty, let me get to that slide here. Oh, there we go. So like, for instance, on that bottom left, or just any of those bottom graphs, right, we plot them, and what we look at is the slope and the y-intercepts of this relationship. The problem is when you have limited amount of motor units, even though we can essentially trace and track the motor units with it being indwelling, we can't really infer too much because one, depending on where the needle or the wire is, it might be too close to the surface or too deep. And so you're really only looking at 4 motor units, and dynamically, it's kind of really hard to determine what's going on. Now, it's been done before, and typically what happens is they'll essentially do multiple contractions over and over and over again and build this pool, but then they'll start to group the motor units, and that may not be the best way, in my opinion, and our lab's opinion, to actually examine how motor units operate. Because chances are you're essentially grabbing the same motor unit again, and now it's placed in a different position, which may like affect the interpretation of that signal. But yeah, you can definitely do it in dwelling, but you're limited to the amount of motor units you can actually observe and assess.
Okay. Well, and just, I suppose, qualitatively, that pattern, and shout out to, right, Elwood Henneman, I believe was his first name. That was what, Alex, in the
65.
50s?
Yeah,
Okay.
there's a train of studies. It was cool because they did it in frogs.
Yeah. So this size principle you defined well. Do you think that this same pattern and the way that the qualitative picture you just painted there and the figures you showed with the MUAPs and recruitment thresholds, do you think that largely holds true for dynamic contractions?
I think so. Yeah, they did some pretty cool studies with macaque monkeys, where they actually implanted electrodes into the spinal cord and then actually in the brain as well, to see like when they're doing dynamic contractions, are they recruiting motor units right in an orderly fashion, or is it kind of like pick and choose? And so it's still kind of up in the air because it's so hard to decompose dynamic signals, right? But I think for the most part, the size principle has like hold pretty steady, right? There are instances where you might get a motor unit that gets recruited, let's say at 70% from the first contraction, maybe gets recruited earlier. And that gets kind of tricky now, because let's say for the first contraction, you were moving really slow. But if you decide to move fast for some reason, that extra excitation that comes from the brain might actually shift that recruitment threshold down, and then you're recruiting a motor unit earlier. Right. But we don't know. And that's the hard part. I know there's a lot of good researchers that are trying to decompose dynamic signals and see if that relationship holds true. And I think for the most part, it still holds true, but there are instances where you'll have different shifts, where maybe the first time that first motor unit was recruited first, and then the second time was recruited after, and kind of flip the flop. But we really don't know, and it's hard.
And correct me if I'm wrong, but is the term motor unit rotation, whenever we're recruiting, is that a controversial term in neuromuscular physiology?
No, it's tricky because, like, in theory, it makes sense, right? A motor unit fatigues, and so another one comes in, and then once that one's good to go, it turns back on, and so on and so forth. But it gets really, really tricky to like determine if that actually does happen or not. And I'm not confident to say yes or no, but in theory, it sounds fun and it makes some sense, but I don't know if we've been able to actually prove that.
And so theoretically, as we are approaching failure, in one example, or just doing a heavier set, let's put some terms on this. We're just talking about the 30 versus 80 study, that we did at Auburn. So very light versus, considerably heavy. As we approach failure, the behavior of those motor units, is it fair to say that the smaller motor units that innervate smaller fibers, probably more of the type 1 variety, do they continue to be recruited all the way up until, let's just call it mechanical failure, where there's intent to produce force, but the limb ain't moving no more. We've reached failure. So do the small motor units stay recruited or continue to be recruited in addition to calling into action the larger motor units that innervate the larger fibers, more of the, let's say, the type 2 variety?
That's pretty much it. Like, that's the beauty of them, is that they're fatigue resistant, right? Or really good at staying and maintain. You can go down the rabbit hole explaining why that happens, but for the most part, you're always going to recruit those motor units, the lower threshold motor units. And with failure and fatigue, you recruit additional motor units, right? When one starts to I'm going to say fail. I don't think the system fails. I think what happens is there's more inhibition to kind of preserve that motor unit, right? You can go and say that there's probably energy issue or anything else. You have your group 3 and 4 afferents that give input to the brain and said, hey, if we keep going, this is known as like the central governor theory, that we keep going, we're going to hurt ourselves, right? But to your point about the 80 and 30%, I have a firm belief that, let's say you take every set to failure with 30%, you're not recruiting the whole motor unit pool. You're recruiting whatever you need to complete that task, right? And so chances are, and there's a study by Muddle et al. And then Miller et al., where they compared a 30% those tracings to failure compared to either 70% to failure and then 190% to failure. And we look at those slopes to kind of indicate how that relationship is between the motor unit action potentials versus recruitment thresholds. So the steeper the slope, the good indication that we're recruiting pretty large motor units, right? The change in motor unit size. And those studies basically suggest that when you're taking a set to failure at 30%, even though you're going to failure, you're not recruiting the whole motor unit pool. You're recruiting maybe up to 50%, maybe 60. But if you do 190% contraction where you're working really, really hard, you're recruiting majority of that motor unit pool. So it's really hard to say confidently when you are training to failure with lower thresholds that you're recruiting everything. But for the most part, those lower threshold motor units are going to stay active and they're going to want to keep working because they're the ones that are going to supply the force when those higher threshold ones can't maintain force or torque output.
Okay. Well, and I remember, with some of your OSU colleagues, this would have been 2017. We talked a little bit about that study. One of the ideas that we had to sort of interrogate this is if we decompose the signal and we have this conceptual idea of which motor units are being recruited that innervate more or less type 1 versus type 2 fibers. We could collect muscle biopsies, and then you mentioned where does the failure occur, sort of. What's an objective measurement that would help to indicate the type 2 fibers, for example, are more recruited in a heavier condition versus a lighter condition? And so we thought about staining for glycogen levels. Mm-hmm. So if you could, if we saw that the heavier condition resulted in a greater recruitment of the larger, type 2 fibers, mm-hmm. So those larger motor units with larger motor unit action potentials, if we stain the same pool of fibers after we do a biopsy, well, theoretically, we should see reductions in glycogen content in the type 2 fibers. Mm-hmm. And that was a very difficult thing to do. And that's why it's not published, because we didn't have confidence in the data. But do you think that is that an interesting research question worth exploring further? Do you think that it is related to reductions in glycogen? Yeah.
I think Stu Phillips talks about that a lot, right? Where if they were to measure glycogen levels, that if they're essentially, for the most part, depleted, that you recruited the whole motor unit pool. I don't necessarily think that's the case. And I think a lot of it has to do, first and foremost, in my opinion, the load they're having to train at or exercise at. I think that's the predominant factor. Because you're only going to recruit what you need to complete a task, right? And then when you start to fail, it could be for, bioenergetic needs, glycogen. But it'd be super interesting, and I don't know how it would be done, because one of the problems that we saw, and Dan can attest to this, is we need them to, when they're doing these contractions, to be as smooth as possible, because when they start to deviate from that, that can affect the sensors or the signal, and then our decomposition is then affected by that. And so sometimes what ends up happening is we end up throwing a lot of participants away because we just can't use their data, at least not confidently. And so anytime motor units are involved, for instance, we're working on a paper right now where I had, I forgot how many people completed the study per each group, but the OOF study that Dan was participating in, right? We did 3 sets to failure. And so we wanted to see, most studies only did 1 set to failure. No one trains on 1 set to failure, right? We do 3 or plus. And so for the OOF study, we did 3 sets to failure. And then another way to normalize that is to compare it to 100% contraction. So that's one way of doing it, but the biopsy stuff will be a lot of fun. It's just super expensive and super tedious. And I don't know. Kudos to anyone that can pull that off, because that's hard.
It was challenging, and we didn't successfully pull it off. So, I still think that would be a really cool study to quantify that. You mentioned afferent nerves. And so for our audience, perhaps it would be good to differentiate between afferent and efferent. In simple terms, afferent being sensory nerves giving feedback to the central nervous system, efferent nerves, what we're really talking about, the nerves that innervate the muscle that elicit excitation, right? Do you think that the afferent nerves, and in some sense the pain threshold, if you will, or tolerance that one has, is one of the primary reasons that failure occurs, or is it more energetic in nature?
I think it's multifaceted in a sense, right? Because they've done studies where they blocked off pain sensors, and they realized that people can actually prolong maintaining a contraction. So they say most of the studies do like a 10% contraction, right? Hold it to failure, and they'll compare even blood flow restriction. And when you block those sensory inputs, people can actually just train or complete that task for a longer period of time, because they don't have any feedback saying, hey, this hurts, we're going to get hurt, so on and so forth. So I think that might be a reason, right? Then you start talking about the psychology of lifting to failure. Some people don't ever actually train to failure because they don't know what that feels like, because they're like, this is heavy, I don't know if I can do 1 more rep. But then you have like someone like Mike Israel yelling at you, you got 1 more, and you're going to knock out that 1 more. But if you were to block those out, not that I suggest anyone blocking them out, they're there for a reason, right? But
is there a supplement that you're aware of that desensitizes afferent nerves we could just take and rip off our patellas on the way to growing quads?
Lidocaine.
There we go. Hey, let's order some.
Yeah.
We do not endorse these purchases, please, guys. It's a joke.
No, Dan, you probably remember this. There was a study where they actually they had rats swimming in water, and they blocked off sensory input, and they found those that had no afferent input swam for some ridiculous amount of time. I think for 24 hours straight, compared to those that actually had input, and they were they gave up after a while because they were just I can't be going.
Yeah.
I don't recommend it. Yeah.
was not, I guess, what am I trying to say here? Anyways, I lost my train of thought.
Yeah.
They didn't-
Maybe
They
What's
didn't
his
want to
name?
live more than they wanted to experience the pain of fatigue, and I think
Yeah.
that's a difficult part of research, is getting people to go to failure. So, like you mentioned, Alex, is they're doing a trapezoidal tracing, and it's all timed, it's standardized, right? Where they'll take, 2 seconds to gradually increase. They're contracting the muscle, and then they're holding 50% for how long?
12 seconds.
12 seconds.
So it's 10% per. It's 1 second per 10%. So if you're doing a 50% contraction, it's 5 seconds going up, 12 to 14 seconds. You're holding it steady as possible, and then 5 seconds coming down, and then you relax.
Right.
Yeah.
And for the OOF study, where we were looking at neuromuscular measures of fatigue during acute resistance training for a leg extension, essentially. It's really hard. So I think if you haven't done it before, especially in a research setting, it's a little different when you're in the gym training, versus having a group of scientists, researchers around you, and you're trying to actually go to true mechanical failure. It's an experience, especially trying to maintain this tracing, because there's typically like a red line, and then you can follow your tracing that's like blue, and you're trying to get a perfect tracing on this trapezoidal shape. And after the first 2 or 3 reps, at least for me, I fatigue very quickly. I think I maybe got 5 on the first set, and then it was like 2 and 1. You're trying to contract, and you can't. And you'll get it up, and then you're not even tracing the red line. It's just coming down. If you're really trying at some point, it's an experience. And it's, to your point, it's very difficult to get many people to get to that point, which can influence the measures that we're collecting, and therefore influence the interpretation of results, not only in this area of research, but other areas, right? Like some of my dissertation work predicting reps to failure with velocity. I think I grill myself in the limitations of that paper, but it's hard. It's really, really hard to get people to go to true mechanical failure, where they are trying to contract, but they can't. And the sensation of discomfort is high. It's just difficult. So I just wanted to highlight that.
I will say it is one of the gnarliest pumps, because you're essentially occluding the leg, because normally during exercise, as you're going through the ranges of motion, right, there's occlusion and, release, normal blood flow. But when you're in that position, it's locked in, you're occluding those 2 areas, and that pump is gnarly. I mean, like the one thing I regret not doing for that study was measuring muscle thickness as a way to measure swelling, because that would have been interesting, right? Because we had 2 different protocols. We had a dynamic isokinetic protocol and then an isometric protocol, because I think they influence or neuromuscular performance is influenced by the type of training that we're doing. But yeah, I wish we did ultrasound, because that would have been cool to add, but you live and you learn.
That'd be a good study, man, just to do like a acute swelling study on that specific measurement, like the responses to that technique. And just to put some more context around that as we dig into this, because we covered a lot of ground, like the intro to and some of the methods already, so we can probably jump into the results pretty quickly for practical application purposes, and in helping to interpret this, particularly the relationship between changes in muscle size and changes in strength. That's kind of where we're headed. The technique you do a maximal voluntary contraction and you quantify the force, right? And so we have this force value, and the tracing, correct me if I'm wrong, is based upon 100% of that value. And typically you trace up to, is it 50%?
I think for this study, they did 70 and 40 for the Pope study. So you try to, and 70 and 40 are kind of arbitrary. Like you could do, actually they did 100% as well. So they're actually able to trace the whole motor unit pool or try to get the whole motor unit pool. The problem is getting up to that, let's say 90 or 100%, and then maintaining it long enough for the algorithm to kick in. And so by like the going up part is kind of hard, but maintaining it, oh my God. I mean, imagine like holding a 1RM in the bottom of the squat.
Right.
12 seconds, or like at 90 degrees, not necessarily the bottom, but 90 degrees. It's brutal.
Yeah. That's an experience too. Yeah. Cause it's all tempoed. It's not just, you can do the just push, right? Like you encourage them and then they just as quickly as they can contract and hold. But when you're doing like a 90 or 100% tracing, it's a tempo up. And I don't think that I was able to get to the top line when I did that because it's just so 80%. It's so slow that for me, where I would have I think a larger MVC relative to the other participants that were in some of these studies, that for people like me, especially I fatigue a little more quickly. It was just that ramp up protocol. I could not hit like a 90% tracing and hold it or 100%. It's very difficult.
That's why we choose 70, and the OOF study was 50, but most studies do 70 because it's like we're pretty confident people can get up to 70 and hold it across. 80 is kind of pushing it, but 70 we're pretty confident that people can do it, even untrained people.
Alex, do you see a big difference in total number of motor units recruited going from something like 70 to 100% MVC?
So one of the issues we have is superimposition. So as the level of output increases, the EMG burst increases, right? So the EMG, I should have prefaced this all by saying the EMG signal is a combination of the motor units that are activated. So the size of the motor units, their own action potentials, right? All laced on top of each other, overlaid on top of each other, and their firing rates, right? So it's a combination of both of those. The problem is as we try to decompose a higher load or higher intensity contraction, there's more superimposition. So the algorithm can't actually distinguish the different motor units because they're all overlaying. Well, one's turning on, the other one's essentially the waveform's coming up, the other one's coming down. So it actually makes the amplitude smaller and bigger. And so it can't figure things out. And so that's one of the issues with doing, let's say, an 80 or 90% contraction is the number of motor units it can detect decreases, right? Because it's only going to be confident with maybe 4 or 5. Sometimes if we're lucky, we can get up to 10, which is kind of a good number we're looking for. The other issue is the recruitment threshold that they're being identified at. So like, for instance, at a 40% contraction, it's really easy. It's really nice and slow. It can actually, in that example, for my study, I think the earliest one we detected was at 5% of the MVC, and the latest was like 40%. So that's beautiful, right? Good spread. But those higher intensity contractions, sometimes you'll get, let's say, an 80% contraction. The earliest one it can detect is maybe 60%. And then the latest it gets to like maybe 70%. So you only have like a 10% distribution. Well, that's not painting the whole picture. And so that makes it very difficult to infer, what's going on because you're only looking at such a small small sample of motor units. You want a good distribution and number. So that you can say confidently, like, okay, this is how the lower threshold motor units are operating. These are how the higher threshold motor units are operating. But when they're all clustered together, you're just like, well, I don't know, they're all on top of each other. So it's hard to distinguish.
So in your opinion, going from 70 to 100%, with the understanding that we're less confident what happens using this technique from 70 to 100%, do you think that the additional force that's produced has more to do with recruiting additional larger motor units or greater firing rates of the motor units already recruited?
Combination of both. I think that it depends on the muscle too. Like for instance, the FDI, you typically see that it stops recruiting motor units about 65% of MVC. And so in order for it to modulate force at, let's say, 100%, it increases the firing rates. And typically what we see is the lower threshold motor units are increasing their firing rates to compensate. Versus the VL, where we can get up to 90% recruitment, right? Meaning that up to 90%, we will see like the latest that motor unit would be recruited. Then maybe that extra, the last 10% will be the increase in firing rates. But for the most part, depends on the muscle. But the idea is that it's going to want, the pool is going to want to recruit additional motor units because those additional motor units have larger twitch forces, which allow them to produce more force. And so that's the best way of explaining. So it's going to want to recruit additional motor units. But when the system starts to fatigue, then you start increasing the firing rates and then you recruit additional motor units. So there's a lot of ways of looking at it, but that's the best way of kind of breaking it down. Depends on the muscle. And then its reliance on recruiting additional motor units.
Okay. Well, and we know a lot about, this is similar to a lot of the biopsy research that I've done. We know a lot about the vastus lateralis, but we don't have a lot of biopsy data from like the traps or the delts or biceps by comparison. A lot of this work is done also on the vastus lateralis, right?
Yeah, vastus lateralis and the biceps. The biceps are really nice and clean too, because they're not pinnated. So it's nice to kind of see the signals because they're, parallel fusiform. So those are the two. We try to decompose some tricep data, but that's hard.
Yeah. Okay. Well, and it's interesting, I guess, to zoom out a little bit here because that was fun for us. But as far as like the practical applications, one example in my mind that's relevant here, especially as we get into the results of this study, you can kind of think of it as loading. So like we talked about different percentages of MVC. Let's extrapolate that for a moment to percentages of 1 rep max. And one thing that struck me during the PhD that inspired our collaboration with OSU was related to some of the data that I came across on, and it influenced the design of my dissertation, why we picked the percentage of 1 rep max that we did. And you see kind of this in the acute muscle protein synthesis research that has investigated the MPS response to different loads. And I understand that there's considerable body of literature showing that commonly measured by ultrasound, if you do 30 or 40% of your 1 rep max, 40% is probably the lower bound I would put on loading here. To failure, you equate volume between like, let's say 40% and 80%. Schoenfeld's done a number of studies. You mentioned Stu Phillips and others. From a ultrasound muscle thickness measurement, you don't see much of a difference in growth outcomes after several weeks of training. In other words, lifting the lighter load to failure or close to it for a similar number of sets as one would lift, with 80%. From an ultrasound muscle thickness standpoint, we don't see much of a difference in change in thickness. So with the acute MPS research, it's interesting that some of it shows this sort of graded response in an increase in MPS past like 30% to 40 to around 60%. You see it kind of plateau, and then between 60 and like 90%, you don't see much of an increase in acute MPS past about 60%, some of the preliminary data show. So I say that to say, so long as one is lifting probably right around that 60% or higher, we're probably okay as far as recruiting most, if not all, of the fibers. If you're taking sets to an endpoint close to failure, would you say that's what's going on? Or are we missing gains by not occasionally doing something like 90% or higher? Do you think that over time, we would see meaningful growth differences at the fiber level if we're neglecting going heavy?
Don't know. It's tricky, right? Because, like, for instance, in my dissertation, I had one group do singles at 90%, and then one group do the same thing, 3 sets for 1 rep, and the other group did the same thing, except they had like 3 additional sets at 45% for as many reps as they can to have like 2 left in the tank. And the relative difference for the hypertrophy group, which is the 45%, they increased by, I think, like 18% relative. This is ultrasound measurements, right? And then surprisingly, the strength group, which only did 3 sets for 1 rep with 5 minutes in between each set, they increased, I think like half of them increased in size, but it was only like 4%. So I don't know, because I do think that there's potential that lifting heavier could add a little more, but how much more? I don't know. The problem is these studies aren't long enough for us to really like determine if that makes a difference. But Dan and I have talked about this multiple times, that I don't think it really matters. Like for me, if I want to get stronger, I have to lift heavier. That's just how it works. And we see plenty of studies. I mean, the Jenkins 2018 paper showed that high versus low load, right? If you're not training with higher loads, you're not going to get stronger, or at least not as strong as if you were to train with high loads. And that was the interesting thing about my dissertation, is that both groups increased their 1RM and BBC similarly, despite one group being significantly bigger at the end of the study, right? The problem is this is an untrained individuals, and it was only 6 weeks, 16 training sessions. But I don't know. I mean, I think a really cool study would be to do that, right? Where you have one group do, okay, let's add in, let's do the 60%, and then we'll do like 1 or 2 sets at 85 or 90. And then the other group just do the 60%. Does that additional load, right? Training with the load where you know for sure you are targeting the later recruited third motor units that are typically larger, and that a stimulus must be provided to activate them. Is that going to cause further growth compared to the group that only did 60%? And the issue is, I don't think there'll be a difference at the end of the day, because so long as you're training to failure or have a really good training session, hypertrophy will probably be the same. And I've always been curious about that. I always wondered, is hypertrophy going to be the same because you have the same number of training stimulus throughout the weeks, right? You have the same 8 weeks of training for both groups. So I don't know if load would play a role. It'd be cool if it did, but I don't know.
Yeah, my hypothesis is that given enough time, which is really important with this, like again, we just talked about that time course of hypertrophy and perhaps this mRNA pool, right? And you've got to give enough time to accumulate those transcripts to eventually translate them. And we just don't really know, like, where is this sort of threshold? And it's probably on the order of weeks with a continuously presented stimulus. So I would say, we may not be able to pick up the small differences unless we took it for probably at least four months, somewhere around that, duration. One, B, or two, the way that it's measured, right? Without like going way into the weeds on that, the ultrasound muscle thickness doesn't give us that fiber level specificity where we could understand what's happening to the type 1, more type 1 variety versus the type 2 varieties. My hypothesis is that if you're occasionally adding in, let's say 85% of your 1 rep max or heavier, and challenging yourself to, if not to failure, which I generally wouldn't advise doing that, with that heavy of a load, depending on the movement. If you're occasionally adding that in, or let's say regularly adding it in, across, again, 4 to 6 months versus someone who's not, only doing 60% of their 1 rep max or less, even if total number of contractions across that time horizon are the same. I think that based on some of the research that you have here, right, that we are likely recruiting more of those larger motor units that innervate larger fibers, which they seem to be responsive
Oh yeah.
on recruitment and production of tension to grow.
Mm-hmm.
I think if we had the fiber type level specificity, enough time, coupling that with some of these techniques, I think we would see a difference over time.
We were originally going to do, sorry, go ahead.
Yeah, I mean, it's a study that I think would be very worthwhile to do.
Yeah. Trevino, actually, my advisor from a PhD, we put in a grant to do basically the same, my dissertation, but in females and collect biopsies. Cause we were going to do myosin heavy chain stuff, right? Co-expression. But if we were able to get the funding, then we were going to get biopsy samples and then try to see if we can talk to Mike Roberts and see if we can do some of that CSA stuff. Right. And I can, speculate on that, but, yeah, that would have been super. If I had biopsies with my study, oh my God, that would have been like, oh, because the cool thing about my study was the hypertrophy group, their motor units, right? The size of the motor units increased. So they had larger motor units because the muscle got bigger, which is similar to what we're going to talk about in the Pope study here. But what was interesting is that the strength group, even though they got stronger and like maybe 4% bigger, right? And like I think half the cohort actually increased somewhat. Their motor units didn't get bigger, but instead they had an increase in firing rates, right? Because the way we produce force is a modulation between recruitment of motor units and then firing rates. And so one of the ideas long ago, and this is the whole premise of my study, was like, well, if someone doesn't get bigger and they get stronger, what's contributing to that strength? And sure enough, the strength group, the motor units didn't get bigger. So the only way to increase strength would be increase the firing rate. So we saw that. And what was neat is, unlike the Pope study where they said that the motor units recruited, I think it was like 40 and above or 60 and above. I think it was 40 and above are the ones that hypertrophied. My cohort, because they were training at 45%, we saw motor units at 25 and 30% actually increase in size. Right. So maybe even just the idea of the training specificity of the training load specificity plays a role too, as far as like what motor units are going to hypertrophy. So that was kind of neat. But yeah, when we give muscle biopsies anywhere, that's awesome, but super expensive. And if anyone wants to do it, sure, let me know. I'm more than happy to collaborate.
Yeah, we need some funding for that one.
So the 25-30% motor units would be the low threshold motor units. And you're saying that the size increased with the action potential. And so we can infer that those are typically the type 1 muscle fibers. Usually, more or less.
It's kind of tricky, right? Because this is coming from a 40% contraction. So technically the 25, 30 and above for that 40% contraction are considered like the higher threshold motor units for that specific contraction. And I think that's where people get kind of confused, right? Because when we think of lower threshold, most people think like, oh, 30 below is considered low threshold because that's the motor units are recruited early on. But it's really based off of the contraction intensity in itself. So for instance, at the 40% level, I would say anything that's 20 and below is considered lower threshold, smaller, typically more type 1. And then anything above that would be more higher threshold, which would be a little combination of type 1, type 2 co-expression. But as you get closer and closer to the 40%, you're looking at more type 2 co-expression, right? So, but yeah, even still, to think that 25 to 30%, whatever fiber types those are associated with the motor units, they grew in size, which is super neat because most studies don't only seen, I would say 40 or greater hypertrophy, which would make sense, more type 2. But yeah, I don't want to confuse anybody, but it gets kind of tricky, right? Because it's based off of the contraction. And then you start talking about relative contractions versus absolute contractions.
Yeah. Well, and your study, right, Alex, was in untrained individuals, correct?
Mhm. Yeah, so
So
they're going to change regardless.
yeah, I wonder if you replicated that study in a highly trained population, if we would see any differences there.
That's where it gets really cool because, in my opinion, I think up to a certain point, neuromuscularly, when I say neuromuscular, I'm talking about like, let's say firing rates, it can only change so much. So the only thing that can really change is the muscle, right? Size. So I would speculate that we would probably see further increase in the motor unit sizes. How much more? I guess it just depends on how much hypertrophy occurred, right? And that's arbitrary depending on the technique you're using.
Mm-hmm. That's probably a fantastic segue. Really good groundwork laid there, guys, into this paper. And Dan, I mean, you can, or either of you, I know you both are very familiar with this. Just a quick overview, and then I think the results are going to be most relevant to explore.
Yeah. Yeah, so I think this was, and correct me if I'm wrong, Alex, the first study that looked at the correlation between the vastus lateralis cross-sectional area measure with the change in the motor unit action potential size in human subjects. Because I think prior to this, there was speculations based on previous research that was done in vitro, where they would have an isolated muscle fiber and do some of the neuromuscular measures with the size of the fiber and make inferences on that. But this was done in humans. This was a resistance training intervention. And so a lot of the methods that we just talked about for the last 45, 50 minutes is pretty much what they did in this study. So I've got it pulled up here on my screen and I'm just scrolling through. I'm going to scroll to the methods, and first thing is the study design and experimental procedures. So the population is always kind of a good place to start. Like who did we sample from? Because that's always very relevant. And so they had 20 young, as they defined, recreationally trained, which is, I think, a vague term in the, because what does that mean? I think that could be a wide distribution of resistance training experience. It's kind of like a catch-all term that's used in the literature. And when we don't report resistance training experience, what they show here is resistance training experience was greater than or equal to 6 months. So they at least had half a year experience in doing something in the gym. But I would guess, knowing OSU, having, been a part of resistance training studies, that you get a range of experience, even when you're looking for resistance trained individuals. In the paper that we published, just the validation reliability study with the Rep 1 device, that was one thing that we did, that I ensured that we did, was define what we meant, that we used the term recreationally resistance trained, and then we defined what that meant was that these were participants that had been training continuously or consistently for at least a year, performing the squat and the bench, which were the exercises used in that study, in our study. But they weren't competitive athletes. And then we gave some descriptions on what that was. So they wouldn't be, I guess, elite level lifters or advanced. So we tried to define that. So I just wanted to, I think, start there as they use this term recreationally trained, which is a little vague. But I think we can be confident that these were resistance trained to some extent. So these aren't untrained. This isn't an untrained population. And these are all male participants. The mean age of this group was 22 years of age. And they performed this resistance training for 8 weeks. They did 3 sessions per week, and they didn't, as far as reading through this, disclose the exact exercises that they did. But what they say is that the training sessions consisted of 3 sets of 10 repetitions for 7 dynamic resistance exercises, 3 of which targeted the knee extensors. So not entirely sure what exercises were used. I would speculate that it was a squat, leg press, and leg extension.
Got it.
More than likely. You probably confirmed that from Hester and DeFreitas. So, and they also did quite a bit of set volume. So if we think about what's been talked about is the set volume for muscle groups. So this is a little side note, is that they performed 3 quad exercises, knee extensor exercises, 3 sets each. So within a session, that's 9 working sets, assuming that this was 10 repetitions to like a rep max, a 10 rep max. That's 9 sets per session. And then within a week, that's 27 sets. So that's on the higher end of the direct set volume for this muscle group. So I think that was just kind of interesting to point out. And as we'll see when we get to the results, they grew quite significantly per the measurement utilized in this study. So anyways, moving on, they did the motor unit. Go ahead.
And the loading is not clear, right? The percentage of 1 rep max or proximity to failure.
Yep, yeah, it just says 10 repetitions.
If I'm not mistaken, I think they trained, maybe I'm thinking of a different study, but I think they trained from like 65 to 85, like it was linear periodization. But I could be wrong. If it's 3 by 10, then it was probably the same percentage the whole time then.
Yeah, I'm not sure.
I'm thinking of a different study. You're right.
Yeah, they didn't disclose some of that information. So we have to make some cautious inferences from this.
But based on other studies from the lab, that's a reasonable guess, right? That it was probably between 65-85% of their 1 rep max.
Most likely.
I mean, Alex, to just add a little context, do you think it was to failure or not?
I don't know. For that specific study, I don't know. I think they only went to 10 reps. But sometimes I feel like training studies, and this may not necessarily towards post paper, sometimes you'll have some people that do more than 10 reps and less than 10 reps, right? And it's just up to the reviewer or researchers to adjust the load accordingly. But for the sake of argument, I would assume they got pretty close to failure.
Yeah.
I think. I don't know. 100%.
Within a reasonable proximity to failure was likely. And whether, the reasons that things are included or not included in a research paper aren't always up to the authors. So I think that that's also just important to say that this, when you're publishing, it goes through a review process. And sometimes things are, asked to be removed or not included. There is sometimes word limits. So, I think there's just different reasons why some information is included in some studies, some aren't. But anyways, moving on, the next thing is the motor unit recordings and processing. So we've talked about this in pretty good detail, Alex, for the first hour. So they did the trapezoidal tracings, and the MVCs. They did multiple percentages, I believe. But per this graphic, I think it's figure 1. They have percent MVC. They're showing an example of a participant's tracing. So it looks like this was a 100% tracing. And we can see that it looks like this individual didn't quite get there. This could have been me. It wasn't, but that's typically what mine would look like. So they do this trapezoidal tracing, and then figure 1B, they have the motor units, 1, 5, 10, 15, 20, 25. And can you explain those numbers again?
You talking about the recruitment threshold at the bottom?
Yeah, so looking at figure 1B, what do those numbers signify? They probably say it right here.
You mind sharing your screen?
Individual motor unit spike trains generated from the decomposition of the surface electromyographic surface EMG. Let's see here.
So I had a paper.
I think it's just the ID, right? It's just the unique ID for that motor unit.
I'm assuming that it looks trapezoidal, so it must be
Okay,
low to
so
high
you said B?
thresholds.
Yeah. So that's the specific motor unit. Yeah. So it's the motor unit number 1, motor unit number 5, and motor unit number 10, and then those little bars next to that are the spike trains, so the firing rates.
Okay.
Yeah.
And there's a process when they're collecting these signals and decomposing. There's like a quality check, and they talk about it in this paper with the motor unit exclusion criteria analysis.
Mm-hmm.
Can you talk a little bit maybe about that?
Yeah. So it's the PD3 algorithm. It's I believe it's called the where's that? The test retest. And so what they're doing essentially is within the motor unit firing rates, they are putting in the motor units, looking at the firing rates, and then kind of reversing it. So they're kind of testing to see, are those firing rates legitimate? And if there's too much variance between the spike trains for that motor unit, it says that this isn't a real motor unit. Does that make sense? So let's say a motor unit pulses every second, right? And then they shift a little bit and they find the same motor unit, and they look at those pulses, and this time it's like 1 every 2 seconds. And it's like, we can't be confident that this is like a true motor unit because the firing properties are not making sense, right? So then it kicks it out. And then it goes to the next motor unit and it does the same thing. And if it passes within a certain validity of, like, I believe in this case, 90%, then it keeps that motor unit. We can go down a rabbit hole of like what people think about this. So what we do in our lab is spike trigger averaging, where we actually look at the average of this firing rates and compare from the raw EMG compared to what the DELSYS, which is this software, is doing. And then if there is a good correlation between those firing relationships, then we keep that motor unit. But this is before this paper came out, before we had to do all that stuff. But yeah, it's just a way of verifying if it's a true motor unit or not.
It's kind of like a quality check. And then if it passes the quality check, that data would then be included in the analysis. And that's an important point, because they started with 20 participants and they ended up only being able to use 10 of the participants' data. So they started with 20, did all of this work to do a quality check, and can't use it, which is unfortunate.
And that's what I mentioned about earlier. It's just like you have to have such a huge sample.
Right.
Even with the biopsies, right? Let's say you were successful with getting biopsies pre and post. Well, there's no guarantee that the motor unit data, and then Cody, I think this is kind of what you experienced, and there's no guarantee that motor unit data is going to be usable.
Mm-hmm.
But now you
feel
just
it
have
on my soul.
Yeah. Oh, it's a pain in the butt, man. But it happens.
Mm-hmm.
A lot of researchers can relate to that. A lot of work to lose a lot of data that's very valuable, and that's just part of it. So, like we mentioned earlier, a lot of studies use the vastus lateralis for their measurements or area of measure. So that's what they did here, was the ultrasound measure, and they looked at the muscle cross-sectional area versus muscle thickness. And I don't know if they have an image. They do. So figure 2A, this is kind of differentiate between the CSA, or a CSA measure, versus muscle thickness. Would you mind maybe explaining, either one of you, I guess, feel free to jump in, explaining the difference in measuring CSA versus muscle thickness? Just maybe like the technique, and then kind of how you would interpret that.
I'll let Cody talk about it, since I've been talking about motor units this whole time.
In this case, this was an ultrasound-based assessment.
Mm-hmm.
Yeah. So, I mean, we talked about cross-sectional area before, but it's you take this three-dimensional muscle, right? We're turning it in different angles. And so if we look in cross-section, we're looking at the area in cross-section, whereas from the surface, looking at thickness, it's getting at the same idea, just different dimensions, a different angle of sight, basically. And, with ultrasound, as long as you're careful, I'm less concerned about agreement between those two, right? Because if you got it in the very, very similar region and we're looking at this way, right, from the surface versus trying to get at, the area. Does that make sense?
Mm-hmm.
So your transverse versus sagittal plane.
Yeah. I think they could have perhaps just stuck with one, but it's cool that, both are in here getting at, change in size, right?
Yeah, I think they just did cross-sectional area.
Okay.
So muscle thickness wasn't in here. I just wanted to differentiate that because sometimes you'll hear CSA or muscle thickness, just differentiating that those are two different types of measure that are similarly related.
And CSA is certainly more difficult to measure accurately. Yeah. And then you have to do the image analysis, which is also, you got to be careful with that part too. Right. Not just capturing it, but then analyzing it. Yeah.
Yeah. And Alex, I think you've used ImageJ. That's a very common software where you get this image from the ultrasound, you download the JPEG or whatever image file, you drop it into something like ImageJ, and then you're able to essentially, you can set the, what am I trying to say? The measure, like you can standardize the measure. So typically on a ultrasound, right, we would have like the 1 centimeter at the bottom. Yeah, we would draw the line as 1 centimeter, and then you can standardize the measures off of that in ImageJ. And then you're literally taking your mouse and clicking from the top part of the muscle tissue to the bottom. And there's potential human error in that. And then just with the ultrasound itself, right, this was enlightening the first time I used the ultrasound, and how I then started interpreting studies was how much, like with just the muscle thickness where it doesn't move, that the amount of pressure you push on the muscle, how much that can really change that measure quite significantly with not a lot of pressure. So if you're looking, there could certainly be a lot of error in that if you push a little bit harder and then you're going a little bit more gently to where it would magnify the post ultrasound measure if we're doing a training study. And I think that's why people are such big proponents of, like double-blinded, a randomized double-blind study where you could, if you could logistically do this, which we can talk about that briefly, you would have somebody that's blinded to, in your case with the dissertation, there was 2 groups. You had a control group too, but you had 2 different training conditions where we would want, if we were doing a randomized or a double-blinded study, that the person doing the ultrasound would be unaware of who's in what condition, so that they wouldn't be any kind of bias wouldn't influence how they're measuring that pre and post. And then once you have that data, is that those images could then go to, it could be the same person because they wouldn't have any idea, or go to another person that could then measure them, and they wouldn't know which is what condition. It would just be coded. And then that data could go to another person in a spreadsheet that's got a unique identifier, and then they would run the analysis, and it'd be like, these are the results. And then somebody would have the key that would, like a master key, that would identify this unique identifier is in this group, and this is this group. But as great as that is, there are just logistical limitations to being able to pull that off, right? So one is, in your case, it was basically you running the study, right? Like having somebody that can be available, that's trained to do this assessment, like the ultrasound. That's not always logistically possible. As much as we want to do those things, I wasn't always available to do that. Or some of our other lab mates like Steph, like they had their own studies going on. We're PhD students. We're trying to get this done. So at the end of the day, if you don't do it, it's not going to get done. And you have people that are reliable, but they're not always available. Right. So I think that, yes, we always want to try to do things like that, but it's just not always possible. I guess I don't want to say it's not possible. It's just, it's much more difficult when you're on a time constraint, when you're a year and a half out from graduating. You know, there's just logistical constraints with that. So I just wanted to talk about that real quick. But moving on is the statistical analysis that is utilized for this type of research, which you can speak well to this, Alex, is they typically do regression. So when you talk about slopes and y-intercepts, and you had a nice figure from one of your studies that you pulled up at the beginning of this talk, is in this study, they ran both linear and polynomial, quadratic, so nonlinear, curvilinear regression models that best fit the data. And then they explained how they determined the best fit model. There's different ways you can do that. I didn't read their reference. It was older. You can do things like Aki information criterion or stuff like that. I'm not sure how that would pertain to this type of research, but essentially you run a regression model, right? Because you've done this contraction. And is this done on the slopes? So you have to, I'll let you speak, but you have to basically do regression individually for everybody and then extract their slopes and then run regression on that, like a group regression on that.
For sure. Because like what you don't want to do is you don't want to group all the motor units, right? Because it's hard to make not only within group comparisons, but between group comparisons. So what we do is we do linear regressions where we take, in this case, the motor unit sizes and then regress them to the recruitment thresholds. So we look at the slopes, right? In this case, the slope is the rate of change in the recruitment, and the y-intercept is the size of the smallest motor unit, right, recorded. And so what we typically do in this study is we take those slopes and then take the pre and post measurements of those slopes and then make statistical analysis that way. There are more appropriate ways to do statistical analysis. So I believe if there is, or there are interactions between the slopes, you have to do different follow-up assessments. But I think this was done before we knew what to do. But typically it's the same thing as just looking at the pre and post means in this case, right? In this case, it's just looking at the slopes and in general.
Mm-hmm. Okay.
Yeah, I guess because that's going to be probably an important delineation here. The slope of what exactly? So on the X versus on the Y.
So the X would be the recruitment threshold. All right. So when that motor unit was recruited. So I'm going to share my screen really quick so we can actually see what I'm talking about. All right. So in the bottom here, the bottom X is the recruitment threshold. And then on the Y axis is the size of the motor unit. And as we can see, the early recruited motor unit typically is smaller. And as we recruit additional motor units, the sizes of the motor units get bigger. And for this study, they were basically looking at the slope. Let me move my mouse, sorry. They were looking at the slope on it and making comparisons between pre and post. So in theory, if the motor units got bigger with training, the slope would become steeper, be a greater value. And And there's a lot of things that can affect the slope, right? It's whether it's the higher threshold motor units got bigger, so it become more positive, steeper, or let's say if the lower threshold motor units hypertrophied or got bigger and the higher threshold ones did not, then that tail end would increase, which would cause the slope to get smaller.
Mm-hmm.
Does that make sense?
Yeah.
Yep.
And so for this study, they were basically seeing, okay, well, let's look at that relationship between the size of the motor units and the recruitment thresholds to get that slope and determine if the motor units did in fact get bigger in one way or another, whether it's a higher or the lower threshold motor units.
Yeah. And if you
Right
just
on.
leave this up for a second, just for anybody that's watching this, is the slope is when we do regression, we get an equation, a y equals something. And so for bivariate regression, linear regression, I think we've all been exposed to the y equals mx plus b, probably during grade school at one point, and we were like, never going to use this again. Boy, was I wrong. I have used this equation a lot. And so we get y equals mx plus b. Again, this is bivariate. There's one independent variable on the x-axis, and then you have a dependent variable on the y-axis. And you get y equals mx plus b, and the slope would be in this equation, the m, right? And then x would be where you would, depending on what you're trying to do, you could plug in the x variable and then add or subtract the y-intercept depending where it intercepts, would be that individual variable there on the equation at the end, the b. And so what you're doing is you're running this regression equation and you're pulling the slope value out of it.
Yes.
Yep. Pretty much it. Yeah,
And
that's it.
okay. So I just wanted to clarify that. And so with this study, make this smaller. You're good to stop sharing. They ran the regressions for each motor unit. Is that correct?
Yep.
Like on the slope? Or I guess that's
So
where,
each of those dots,
because they have a figure where they make a nice table.
yeah. So I'm trying to
I say figure,
see.
table. Table 1.
Yeah, so table 1 is showing for each subject the number of motor units they recorded, right? And then the recruitment range. So the lowest, for instance, subject 1, the lowest detected motor unit was at 11.4% of their MVC, and the highest was 83%. This is a pre. And then at post, they try to, what's really important for us is in order for us to make pre and post comparisons, so the recruitment threshold ranges have to be pretty much dead on the same, like maybe 5% difference. Because if they're too far different from each other, there's no way of really comparing the lower threshold motor units to the higher threshold motor units, right? You want to have as much overlap as possible.
And you're looking at the recruitment threshold, this RT range.
Yep.
And you're saying that value, that range has to be very similar from pre to post. Because if we look at subject 1 in this table, they look quite a bit different, at least on the upper range. The top value, like pre was 11.4 to 83.8, and then post for this subject was 9.8. So that's similar, but then it was 58.3.
Yeah. And you'll notice what's interesting is that the action potential versus RT slopes are the same, right? And that could be the fact that they didn't have motor units from 60 to 80, right, that were detected for the pre-measurement, which are the ones that are most likely going to hypertrophy. So that slope value could have been bigger, way bigger. Right. That's why it's really important to try to match up those ranges so that you can infer accurately. Well, let's say motor units recruited at 75. Let's compare that 75% recruitment threshold to the 75% from pre, and then make comparisons of the motor unit size from there, which some studies do and don't do. We try to do that because we want to be as like transparent as possible with our inferences. Because sometimes too, if the range is too small, that's going to inflate that slope and make it way bigger than it really is. or smaller, right? In any direction, really.
So is it on this one? It's important. What is it like the APRT slope?
Mm-hmm.
Can you explain that a little bit?
Yeah, so that's just the slope, right? So the change, the slope of the equation, right? So for each increment, or each motor unit, as you recruit motor units, is the kind of the difference in sizes between motor units.
Mm-hmm.
That makes sense. So motor unit recruited at 5 versus 10%. What is the difference? Just like a normal slope, right? Rise over run.
Right.
And so that slope is really showing how much of a difference there are between each recruited motor unit.
That slope pre to post, we would expect it to change.
Mm-hmm.
So that APRT slope after resistance training, if in this study they're looking at changes in muscle CSA with the motor unit action potential, assuming that the action potential gets bigger when the muscle gets bigger, that slope, that APRT slope should change. It would get steeper, right?
It's steeper. Yep.
Another way of saying it is for each unit increase in recruitment threshold, if it's a positive slope, there would be an increase in the motor unit action potential size.
Yep, that's pretty much it.
And if the slope at pre was lower than the slope at post, then the indication would be, well, for each unit increase in recruitment threshold, we're seeing a larger action potential size, so the motor unit got bigger.
Yep. Simple terms, yes.
Right on.
Yeah.
Right on. Yeah.
And so at the group level, there was a positive increase for the slope. So it increased by 0.0013. So that's a pretty small change, but maybe for this area of research, that's large.
Yeah. We got to think about it, right? These are the size of those motor units, and they're measuring in microvolts. So very, very, very small measurement or size. And so, yeah, it may seem like a very small number, but it's pretty big if you were to zoom in,
right?
For the most part.
No, that makes sense. And so then I was hoping they had a figure. I thought they did, but they didn't, as far as a relationship between the change in CSA and the motor unit action potential. I don't know. Oh yeah, they do right here. Figure. I thought I saw it. Yeah, 3D.
Yeah, 3D. Yeah.
Yeah, and so thought I did. I was looking for it. So this is kind of what they were trying to do, is to use a non-invasive neuromuscular measure to detect a change in the muscle CSA. And based on this data, they did. So on the x-axis, they had the delta score for CSA. So that's important for regression, that you use one observation for one participant. And then on the y-axis was the change in the APRT, or the action potential recruitment threshold slope.
Even.
And so they found a pretty strong relationship here. And so you get not only the y equals mx plus b from regression, but you get the correlation coefficient, Pearson's R. And so that's just kind of a general, it's like the effect size for regression. It's the strength of the relationship. And then you can also get the R squared value, which is the correlation, coefficient of determination. And so that's what this R squared value is. And it's basically the amount of variance that X explains about Y, or the amount of variance that the model is explaining. And so we could put that as a percentage. So 83% of the variation in the action potential recruitment threshold is explained by a change in muscle CSA. So these muscles got larger, and a lot of that variation in the change in that motor unit action potential size was explained by a larger muscle, essentially.
Mm-hmm.
Well, that's interesting.
Yeah. And I think the coolest thing about this study was, for the most part, we understood that a larger fiber typically has a larger action potential, right? That's a pretty good correlation between the motor unit size and then the fiber size itself, right? The specific fibers it innervates. What was neat about this study was they did 100% tracing. And so they were trying to get the whole motor unit pool, because sometimes too, and I've seen it in some studies where they only do like 70% and they don't see the same relationship between the changes in those slopes in relation to the changes in. But when you're doing a 100% contraction, you get the whole motor unit pool. And with that, you get those extra higher threshold motor units that are most likely going to hypertrophy, right? And so if you're not getting those, which is something we didn't do in my study, we didn't do the 100% contraction because it's really, really hard. I mean, you saw that out to 80. But nonetheless, the nice thing about this is it kind of supports the notion that when you have hypertrophy occurring, it's typically those higher threshold motor units that are going to hypertrophy. And the larger that change in that slope, a good indication that those later recruited motor units are getting bigger, which is going to slope to become steeper.
Yeah.
And so if you look at figure 3C, they plotted out the regression for the pre, and see it's kind of somewhat shallow for the most part. But then for the post, they had two lines. One where there's small, similar to like pre, right, very shallow, and then an exponential line, which is why they did like the polynomial, regression stuff. And that breaking really is indicating, all right, at what point do we see hypertrophy or these motor units getting bigger? And it's really occurring everything above 40% of their MVC, right, are recruited during contraction, which is awesome, right? That's where we're really seeing the hypertrophy occurring, is for those later recruited motor units, versus like the early recruited ones. Even though the early recruited ones are always being recruited, right? They're always active. It's those later recruited ones that have more type 2, expression that are experiencing that hypertrophy.
Yeah, and Cody, you kind of circling all the way back, you mentioned that you would argue 40% as the lower end of the loading spectrum for muscle hypertrophy, and some of that was the MPS response. Right. So I think there's almost some evidence to support that here too, right? If I'm understanding this correctly, is that around 40% of an MVC is where we start to see the action potential size increase quite significantly.
After training, yeah.
And so that's maybe where they're starting to recruit they are starting to recruit larger threshold motor units that may be more sensitive to resistance training stimuli.
Yeah. So Trevino has a paper out where we cited in my soon to be submitted paper, where even like around 40%, at least for the VL, 40% has a good amount of type 2 co-expression, which would make sense why we're starting to see that hypertrophy occur. And specifically in my study, where I only mentioned 25 and 30 because those are like the motor units with the average range, where we see from like, I think it was from 10 to 35 was the average high threshold and low threshold ranges. But yeah, it's interesting that the 25, 30, and as well as 35, if we were to include that as well, were hypertrophying. But for this study, they were training, I would assume, like 65 to 85% of their 1 rep max. And so it maybe explain why those, recruitment threshold ranges are experiencing more hypertrophy versus the lower threshold.
Yeah. And I mean, to be clear, I'm not saying you can't grow from lighter loads than 40%.
Right.
Mhm.
But the magnitude of growth over several weeks, I do think would be different if you're going consistently below 40%. And
Right.
one study that I think really did a nice job of showing this is, I believe you say the last name, Lascivious. I may be butchering that, so forgive me. But Dr. Brad Schoenfeld was on this study, and it was within subject design, and it was bicep curls. I believe it was maybe twice a week for like 8 weeks, and they did an ultrasound of the bicep muscle thickness for each arm. You guys know the study I'm talking about?
I'm not
So there was
familiar.
so if we were all in this, there's a 20% condition. So like our left arm, we would have done,
Yeah. Yep.
20% of our 1 rep max
Mhm.
to failure for like 4 sets. And then the other arm, we would have done either 40%, 60%, or 80% of our 1 rep max. Same number of working sets across a week. And after, like I said, I think it was 8 weeks. It may have been 12 weeks. But the larger point is that there wasn't a meaningful difference in change in muscle thickness of the biceps between 40, 60, and 80%. But the 20% condition was about half of the growth.
Mhm.
So that's interesting. Connecting
Right.
these dots.
Right.
And which
then
is-
maybe, go ahead, Alex.
I was going to say that kind of showcases the importance of the load, right? Where even if you are training to failure, there's no guarantee that you're recruiting all the motor units that would, in theory, be necessary for hypertrophy.
Right. Guys, I've got to go. I've got to get a treadmill. You guys keep rocking and rolling. This was a blast. I think we definitely, I would like to get into the strength and muscle size stuff. But I think if y'all want to hit on it, and we could, if Alex, if you'd be willing, we could perhaps have a whole other building off of this strong foundation. We could dedicate a whole conversation to just that topic.
That'd be fun. That's my research interest, so that'd be cool.
But this was great, man. Thank you so much for your time. And like I said, you guys can keep going. I just they just got here, so I've gotta go.
Sounds good.
It was a
man.
pleasure meeting you.
Yeah.
Well, I think we'll wrap it up here shortly. We've been on here for almost 2 hours.
Yeah. Thank you, Alex, so much, man. I'm excited to meet you in person soon.
For sure. Thanks, Cody.
All right, guys. Peace.
All right, see ya.
So
yeah, I guess we've nerded out for a good bit of time.
Yeah, it's a lot to unpack.
There is a lot to unpack, unpack, and impact. Decompose, manipulate.
Yeah.
Kidding.
You tell your story.
That's right. Well, yeah, man, I think this is maybe a good stopping point.
All right.
I think we covered a lot of the big bases here. Definitely appreciate having you on. We'll be doing more future talks. I do like that we intended for this to be short format, and it ended up being long format, which is fine because we got a lot of information, and I think that's very valuable. So definitely appreciate you jumping on. Where can people find you?
Yeah. So you can find me on Instagram. I am Dr. Professor Olmos. No pun intended, but yeah.
I think it's underscore Dr. underscore Prof.
But I honestly don't remember. I think it's Dr. Professor underscore Swolmos. Because my last name is Olmos, right? And I like to consider myself swole, but you get around people.
We'll put it in the description. We'll put it in the
we'll link it.
description.
You can find me on there, or pretty much it. I also am a professor at Christopher Newport University in Newport News, Virginia. I just started here and hopefully putting out some pretty cool neuromuscular research and muscle imaging. So
yeah, man,
that's the goal.
Yeah, you've done good work. Excited to get some of your dissertation work out. Which we'll do another talk on some of that stuff. That'll be fun. But all right, man. Well, hey, that's it. for those of you that made it this far, appreciate you all listening. Hope you enjoyed it, and we will see you next time.
Yeah.
Peace. And I'm going to